Hello and welcome to Astrobiology Podcasting. In today's lecture, we're going to do one of two professional development exercises. And in this one, we're going to focus on the concept of scientific inquiry. All of you, as you move through your careers, are going to have or develop the ability to ask questions, participate in active scientific research, hopefully, and in general, always using that scientific method, whether you're designing experiments or just critically thinking about scientific information that's being presented to you. So in this activity, we're just going to delve into concepts of scientific inquiry and hopefully expose you guys to some of the concepts that where does creativity in science come from and how do you pose a scientific question. So just a couple things that we're going to dig into. When I'm talking about scientific inquiry, what I'm talking about is generating ideas and learning how to ask the right question. It's so important not only just to know what the answer is to the questions, but asking thoughtful questions in science is really how we lead to new information and new insight. So we're just going to talk a little bit about what are the creative processes involved in asking a meaningful science and pertinent question in science, and also how do you go building your skill set in developing a scientific question. And the key thing is we have to practice asking questions. So in this little activity, I'm going to expose you to a few concepts about where creativity comes from, how do you develop a scientific question, and give you a little practice examples for you to work on during this activity. So let's start with talking about where do creative processes come from? How are they involved in asking meaningful and pertinent questions in science? So let's start with two people that have had great ideas in the past. Does anyone know who these men are? The person on the left is someone you might know in some of your science classes. The person on the right is someone that you might know from your art history classes. So if you had guessed Albert Einstein on the left here and Pablo Picasso on the right, you would have been correct. Einstein and Picasso were both individuals. They were contemporaries of each other in the early part of the 20th century. And in that period, that's when they had their best ideas. Albert Einstein with the theory of relativity, Pablo Picasso with all his amazing artwork. And I want you to kind of think about this idea of where did the creativity for some of their inspiration and some of these very profound ideas come from. So I want you to think about the creative processes. Einstein, for example, when he was talking about his theory of relativity in the early 20th century, said, I often got my best inspiration in the bathtub. He just would sit and ponder about these ideas, and they would just come to him in the bathtub, so to speak, or playing the violin. Picasso had all of these different meaningful inspirations. So I want you to ask yourself, do you think that the creative processes to come up with an idea or a new hypothesis like special relativity or general relativity is the same as an inspiration for a new painting or a new way to, or a new perspective? In the case of Picasso, his style of painting was completely revolutionary. So where did those creative processes come from? Are they similar or are they different? And ask yourself that question. So I would argue that actually creativity in both science and art comes from the same place. And I would say they were very similar. And creativity is often thought to come from this concept of cognitive dissonance. Cognitive dissonance is basically when you're observing something and there's an inconsistency between what you're seeing and the current theory to support that. In the case of Einstein, he was going against 200 years of Newton's ideas. And so he kind of came up with his theories of relativity, and they really differed from Newton. And that caused a bit of a clash in his brain and also a clash with the scientific community because he was ruffling feathers and going against that idea. But in science, that's what often has to happen. You have to kind of present that new idea, and it may take a couple decades before that idea is accepted. A big example that's happening right now is the concept of the microbiome. For decades, developmental biologists, animal biologists just refused to listen that the microbes could be having such an important role in animal physiology and animal health. But now, after several decades of research, we now see that indeed microbes are playing a much bigger role in human health than we previously thought. So this cognitive dissonance, it's something, again, when you're seeing something that just doesn't fit with the current dogma, the current theory, and that can be very inspirational for where creativity can come from. So again, just to dig into this example that I gave between Einstein over here making his wonderful face and Isaac Newton here. And Albert Einstein always said, "Newton, please forgive me," because I've pretty much debunked some of your ideas. Some of Newton's ideas, of course, still hold true. But Isaac Newton considered gravity to be a force, and he thought space and time were absolute. And one of Newton's most important contributions to science was his mathematical definition of how motion changes with time. He showed that forces causing the apple to fall, so to speak, is the same force that drives planetary motions and produces tides. However, Newton was puzzled by the fact that gravity seemed to operate instantaneously at a distance. He admitted he could only describe it without understanding how it worked, and it wasn't until Einstein's general theory of relativity was that gravity changed from a force to the movement of matter along the shortest space in a curved spacetime. So what's happening, what I mean by that is the sun is bending space-time, and space-time basically tells the planets how to move. So for Newton, both space and time were absolute. Space was fixed, infinite, unmoving metric against which you could measure the motions of things. And he also believed, Newton that is, that the universe was pervaded by a single absolute time that could be kind of like an imaginary clock somewhere in space, that it was relative, or time was the same for everyone. And Einstein completely changed all this with his theories of relativity. And that's why he once wrote, "Newton, please forgive me, because I'm kind of overturning some of your major ideas." So Einstein's big contribution to this whole thing was that he realized that gravity was the movement of matter along the shortest space in curved spacetime, and that essentially time changes with motion. So depending on where you are or how fast you're moving in the universe, your time perspective is different. So if you have an alien spaceship moving at the speed of life, my life will just pass by in a blink of an eye to an alien that's traveling at that speed of light, meaning our perspectives of time are different. And so basically, with these ideas, Einstein revolutionized physics with this new concept that time is changing as you're moving, or how fast you're moving in the universe. So it was revolutionary, and again, a good example of this cognitive dissonance and how new ideas can overturn pillars like Isaac Newton here of ideas, previous ideas. In addition to Einstein and Newton, another piece of evidence that kind of overturned the pre-existing ideas was the ribosome. We talked about this before when we talked about the origin of life, that there was, for many years, people thought that we knew about ribosomes since the 1950s, and everyone thought it was the proteins that was doing all the major function. But it turns out that it was actually the RNA, and it slowly took a long time for people to let go of this idea of a DNA protein world that came first, and then realize that maybe RNA might have evolved first. So again, once you eliminate all the proteins, you can still get functioning ribosomes as you peel back the layers of this ribosome. So again, this was another example of cognitive dissonance, where they just assumed it had to be a protein DNA world, but as they peeled things back, they learned more, that maybe the first forms of life on the planet might actually have been RNA based. So how do you go about developing a scientific question and idea? So how do you build that skill set? And it's knowing how to ask questions is almost just as important as knowing the answers, because then you can actually start to put ideas together, synthesize ideas. And I love this quote by Robert Frost: "An idea is a feat of association." It's really about putting things together, seeing where the gaps are, and then formulating a better or more improved hypothesis or question. And Linus Pauling, another great quote here, says the best way to have ideas is to have a great idea is to have lots of ideas. So you always need to keep that thinking hat going, because creativity is messy and you're often going to make mistakes, but you're going to make great discoveries in the process. And I'd like to introduce at this point in time in the course Mr. Giordano Bruno, who was actually a Catholic priest back in the 16th century, who was the first to propose that there were exoplanets. He actually saw that there were countless suns and countless Earths rotating around their suns the same ways the planets orbit around our star. And we only see the suns because they're the largest bodies and are luminous. But they're planets. He guessed that there were planets invisible to us because they're small and non-luminous. So it seems rather logical, but using the basic information he had at the day. But he quite angered the Catholic Church there. Even though he was a priest, he was actually eventually burned at the stake in Italy for these ideas. But a couple hundred years later, the Catholic Church apologized and they built a statue for him. But so you have to be careful about your ideas because sometimes they can be rather inflammatory based on the current political or religious climate. But Giordano Bruno, we give him credit for the first kind of postulating and presenting that idea about exoplanets, which he'll pop up again later in the semester. Okay, so this inspiration, you were in the bathtub, you came up with a great idea. Now, how do you start developing it into something that's testable? Is your idea even testable? If your question is, I think there's multiple universes and that they bang into each other and cause a big bang, is that something that you can test? Or is your question, I think this gene is doing this function and so forth? So you make an observation, again, that's messing around, being, tinkering around with materials and so forth, asking a question, selecting which of those questions that might be able to be investigated, designing an experiment, reflecting on what happened, and of course, you answer one science question, you get 10 more science questions. So is your idea testable with this scientific method, so to speak? And if it is, then go for it and you can try and test your idea. So when I'm generating a scientific question or I'm thinking about it, it might just be sitting in a seminar thinking about the research that's presented in front of me, or it's about designing a new project or writing a new grant. I try to use my own creativity or my own observations, logic, and my curiosity. Hopefully those of you who want to go on to research are scientifically curious individuals. And what I do is I think about the following. So when I'm sitting there in my seminar listening to someone talk, I think about the following. All molecules, genes, objects, whatever that person is talking about, they must have arisen somehow. And they must have some sort of function, some sort of property, and they must have at some point end. Some point it degrades or changes into, or cell divides or something. Some point it ends, it ceases to exist. So I ask myself the following, even if I don't have the answers. Is that molecule changing with time in any way? Or is that object changing in time in any way? How did it form? What is its relationship to others? How does this object function? Does it have more than one function? Maybe it's used in different ways. What is this object made out of? How does this object degrade? And from where did these things that it's made out of come from? How did the building blocks come to be, and how did it become part of this object? So these are kind of the things that I do subconsciously as I listen to seminars, as I read scientific papers, as you will do throughout the semester, and start asking yourselves, where does it start? How does it change? How is it related to other parts of that, whether it's a machine or a process or a pathway, a gene pathway? So these are the things that you can ask yourselves, even if you don't have the answers. Okay, so you just ask a question and you want to self-assess. Now we're going to talk a little bit about how can you judge the quality of a question, or basically, I shouldn't say judge, how can you categorize the quality, that type of question? Are you asking something that's a little more informative? Are you asking something that's a little analytical? So when I say good or bad, I mean it's very relative. But how do you think you can judge the quality of some of these questions? And the ultimate goal for you guys as scientists is to start to get a little more analytical, move away from the information questions and dig a little more into the analytical questions. That's what this whole exercise is about, building your skills to ask more deep analytical questions. So when I say something's a good question, what I'm hoping you get from that is that it's leading to an improved understanding. Can we improve our understanding of how that protein works, or how that gene works, or that pathway? An okay question is you just need a little more information. It's not going to necessarily help you improve the understanding. I call these, Me Too science questions or something like that, where you can build a little more information. It might not help you understand the whole thing, but it gives you a better perspective. And bad questions really don't do either. They just might even lead to misinformation. They're not really helping you understand how this process or how this phenomenon is working. So here's a couple examples. Are they quote unquote good or bad questions? Question 1. What other organisms have gene X and Y besides E. coli? How would you characterize this? Would you call it a good question, an okay question, or a bad question? I might suggest that this is an okay question. You're just trying to find out a catalog of genes, X and Y, that are in other organisms. That might not necessarily help you find how it's working in E. coli. But maybe, what it could lead to eventually, if you had enough organisms, you could build a phylogenetic tree, and maybe that would tell you a little bit about where the genes came from and how they function. But I would just say at the moment, this would be an okay question. Question number 2. What effect does the expression of the product gene X have on the regulation of gene Y? This is what I would call an analytical question. In my case here, I would call this a good question because what I'm trying to do is get at how this is functioning. So this question will lead to an improved understanding of the expression of gene X and what its relationship is with the product of gene Y and so forth. So again, this is getting at improved understanding, and to me, according to my little categories here, I would call that a good question. The third question, does E. coli suffer from environmental stress? What do you guys think about that? In particular, look at the word suffer. So suffer is definitely an anthropomorphic term. We're just saying, are they, suffering? Are they experiencing some sort of, are they feeling environmental stress, rather than are they exhibiting high reactive oxygen species or a lower temperature sensitivity or something like that. So in this case, I think it's not necessarily a bad question in the sense of the objective of the question. I just think it's poorly worded, and here's a case where you might want to change out that word suffer and substitute in something a little more specific. So here's where a case where you want to be a little bit more scientific in how you organize or articulate that question. So now what I want you to do is make sure you have the Word document, the exercise activity that accompanies this lecture series. And it's a Word document that has a few questions that I'd like you to address and answer. So I'm starting with this painting called Guernica by Pablo Picasso, because I think that you probably each have more experience visualizing and working with art than you do maybe with different scientific techniques and so forth. So I want you to stop the podcast, think about what questions that are coming to mind as you look at this painting. What are you thinking about? What does this represent to you? And it can be, and at this particular exercise, you can think about it as an art historian, you can think about it as a scientist, but think about 3 questions, and there is no wrong answer with this exercise. It's all worth points. If you complete all the activities, you'll get full points. So the only wrong answer is a blank answer. So think about 3 questions, and then I want you to self-assess each question. Do you think that question is informational? Do you think that question is emotional? Or do you think that question is analytical? So write that down on the exercise sheet and self-assess. And it's okay if you might find that you have more, because it's a piece of art, so it's trying to generate emotion out of its viewer. So it's okay if you have a range of different categories here with this particular exercise. Okay, so now in exercise 2, we're going to look at something more science-derived. So here's an image. Tell me about this image. What sort of questions do you have about this image? And if you have a guess of what you think it is, go ahead and put it down on the exercise sheet. But ask the questions and then do the same thing. Ask yourself, is that an informative, analytical, or emotional question? If you get emotional by looking at these pictures, that's fine. Science is also about getting your curiosity, inspiring you to be more curious about things. So that's fine too. But just write that down in exercise 2. And pick one of these. Continue on with exercise 2 and pick one of these, either B or C, and tell me a little bit more. Again, ask some questions. I mean, to me, these are very beautiful. They're all scientific images, but they're very beautiful and can pass for art any day. So tell me a little bit about these images and the questions that you want to ask about that in the activity worksheet. You only have to do one of them. So in this next section, or exercise 3, I want you to listen to a supplementary science podcast that I've attached to this lecture. And I want you to practice asking questions. Just imagine you're listening to this little mini seminar. It's about 8 minutes long. I want you to practice asking questions based on the material that the presenter, which in case is me, and I don't want you to make assumptions. Don't just assume what I'm telling you is accurate, because I'm intentionally misstating stuff because I want you to pick it apart. I intentionally want you to observe and listen to what you're hearing and see if it matches up with the data you're being presented. Then what I'd like you to do is to complete the worksheet. You can then submit it through the Canvas website, and then you'll get points. And again, the only wrong answer is a blank answer. But listen to that podcast. Think about those questions that we talked about before. And again, don't make assumptions. Don't just assume what the person telling you, the scientist presenting their data, is telling you everything, or maybe there's holes in their information. And then so when you're working on the activities, what's the first question that pops into your mind as you're listening to that podcast? And the big thing to always be mindful of is the data supporting the conclusions. And then what are the missing pieces that I haven't told you in this podcast? Is there something that you think should be done next? What would you do if this was your experimental project? Now, I know that this subject matter might be out of your expertise. However, I want you to think about it. I want you to push yourselves and take risks, because that's how great ideas come from. And then at the end of the activity sheet, there's a little survey to see if any of these ideas or these concepts are new to you. Is this just old hat to you? Have you learned anything new? Is there anything you would recommend changing about this activity? So feel free to give me your feedback. We've used this feedback before, and we published a paper that you can take a look at some of the other students' results from past years if you are interested. That's also in this module. But I appreciate any feedback and positive or negative suggestions, things that we can fix. That's all encouraged. So good luck, and thank you for participating in this scientific inquiry exercise.